Okay, this is chapter 2 on overview of animal diversity. So last lecture, well, the recorded lecture I just recorded, I pronounced something the incorrect way. So let's start all over because they gave me an insecurity. So let's move on. So how do animals differ from other life forms? Well, first, animals must consume other living organisms to meet the nutritional needs, so they're heterotrophic. So an animal must eat or ingest or however they consume energy, they have to consume another organism to meet their energy demands. Another thing is that animals' cells are multicellular, meaning that these cells have proteins on their cell membrane, allowing for cells to communicate to each other. So this is what really is special about animals is that they have cells that plant cells also communicate to each other. But there's different tissues of animals that there's different tissues of different types of cells and they all respond to each other, communicate to each other, stick to each other, and basically form one complete organism, making it an animal. They have a muscle and nervous tissue, so they have muscles for coordinated movement and a nervous tissue to execute those coordinated movements. Reproduction and development, whether they go through a larvae, through metamorphosis, which we'll discuss in the next lecture, and they must reproduce. And remember that the animals are dominated by the haploid state. So here is a zygote, animal zygote. It cleaves again, meaning it just goes through mitosis. There's an 8-cell stage, cleaves again and again and again to become a blastula. The space is called a blastocoel, and this is where the coelom forms. So a process called gastrulation is where the cell wall, not the cell wall, the cell membrane of the blastocoel, the blastula, folds in like this. And when it folds in, it creates two spaces, one here and one here. The inner space is called the archeteron, while the outer space is called the blastocoel. And this is where the three germ layers arrive. The three germ layers are ectoderm, endoderm, and mesoderm, which we'll discuss right here. But first, let's just talk about the blastocoel, the blastula, for a bit longer, only 10 seconds. So the opening of the blastocoel, the blastula, is the blastopore. So the ectoderm is the outer layer, and the endoderm is the inner layer. And the space is the blastocoel. So the origins of multicellular animals. So biologists have identified 1.3 million living animals. There's far more to estimate, of course, and the common ancestor of all living animals likely lived about 800 million years ago. So the common ancestor of all animals is a group of proteins called choanoflagellates, and they are basically part of a sponge, and we'll discuss that in the next lecture. And the origin of multicellularity requires the evolution of new ways for cells to adhere, attach, and signal and communicate to each other. So animals are multicellular. Okay, that's that. There's no exceptions. And they're multicellular, meaning that they have signals, whether it's through proteins or chemicals, to communicate to each other. Because tissues have to communicate to each other for the organism as a whole to maintain homeostasis and balance through the environment. So choanoflagellates and animals have sequence similarities in genes involved with adherence and attachment. So let's talk about the Cambrian explosion, which occurred in the Paleozoic era. So the Cambrian explosion marks the earliest fossil appearance of many major groups of animals. So a lot of the fossils today that we find are basically from the Cambrian explosion. So most of the fossils from the Cambrian explosion are bilaterians. Bilaterians. Let's say that. So they have a bilateral symmetric form, a complete digestive tract, one-way digestive system. So meaning that they have a mouth, a separate opening, which is the mouth, and another opening called the anus. We have animals have a one-way digestive system, which food just goes in through the mouth, goes to the esophagus, through the stomach, then into the small intestine, I believe, or is it the large intestine? Well, either one of them, then out through the anus. So it goes one way. So here's a picture of what the Cambrian explosion should have looked like. And there are several hypotheses regarding the cause of the Cambrian explosion and the decline of the Ediacaran biota. So one of them could have been a rise in atmospheric oxygen. So oxygen could have benefited animals or organisms with higher metabolic rates or larger animals. These larger animals then started to eat other animals. And those animals that were being eaten had to evolve to defend themselves or counterattack the predator. And then the predator had to evolve some way to counterattack their defenses or cripple their defenses. And it just kept evolving side by side that they created new species and diversification. And the evolution of the Hox gene complexes, and the addition of new microRNAs. So microRNAs regulate gene expression. They tell which genes in the genome to be expressed and which genes to be turned off. Hox genes are genes that direct development. You'll find Hox genes in places called homeoboxes. So every animal has a set of homeoboxes with their unique Hox genes. And these Hox genes direct development. For example, the Hox gene will tell the body where to grow my thumbs are at, or what should be on your hand. So if somebody messed with these Hox genes, my thumb will grow where my toes are at, or where my eye is at, or where my ear is at, which we do not want. So Hox genes direct development. And the evolution of these Hox genes led to new body plans, so new body parts and different types of body forms. Also, microRNAs led to different body forms and body plans. So animal diversity continued to increase through the Paleozoic era, but it was punctuated by mass extinctions. So here we have the Cambrian explosion, and here is a table of diversity of animals and number of families. Of course, we are talking about biological classification, so families. So here we have about 200 families, and then something happened, extinction, rose up again. Here we have a mass extinction, which is a volcanic eruption, destroys diversification, kills a bunch of species, or a bunch of animals, things like that, and then it just starts again, starts picking it up, starts to recover. Then 5 different mass extinctions. So just to pull out, what can we pull out from this is that mass extinctions end diversifications and start new diversifications. So just remember that in the back of your mind. Like we said, animals can be characterized by body plans. So body plans are a set of morphological and developmental traits. Some body plans have been conserved, meaning that once evolution has done something, it cannot go back. So if I just had I don't know, let's just say a 7th toe or a 6th toe, 2 extra toes, evolution cannot undo that because evolution cannot perfect things. It cannot undo things. So that's what it means when it has been conserved. And others have been changed multiple times over the course of evolution. As you see, evolution changes animals and how they look like. For example, Animals, a population that's black rabbits will survive more than white rabbits, and therefore they change their body plans to predominantly black rabbits. That's just a simple term. I don't want to spend 10 minutes talking about this one point, but that's what you can pull out of it. Symmetry. So animals can be categorized according to the symmetry of their bodies. So this plot here, you can cut it on the X or Y axis, and you get 4 equal parts. That's radial symmetry and bilateral symmetry. You'll cut this lobster in half through one axis, and you get 2 equal parts. So bilaterally symmetrical animals have a dorsal top side and a bottom side, a left and right. So they have a top and bottom, left and right, and front and back. They also have sensory equipment such as a brain and nerves concentrated in the anterior end. This is either called a ganglia or a brain. So this brain allows bilateral symmetric animals to have coordinated movements, meaning that they can look at things, they can be more sophisticated in their movements. That means they can move more quickly, more accurately. While in radiosymmetrical animals or organisms, I'm trying to find my page. Oh my God, I can't find my page. There you go. While radiosymmetrical organisms have a top and bottom, but no front and back, left or right. And they are usually immobile, so they don't move. And if they do move, they're probably just floating in the water, letting the environment move them. They're not actually moving. So tissues. So we all know what tissues are. If you're in biology and you don't know what a tissue is, well, I suggest you should catch up. So a tissue, just a quick recap, a tissue is a group of cells different from another group of cells, whether it's heart tissue, epithelial tissue, arterial tissue, whatever tissue. It's just a group of cells that are characterized differently than another group of cells. And these tissues must interact with each other. And this is why animals have tissues, and all those tissues cooperate with each other. All those cells cooperate with each other in order to respond to their environment, in order to maintain homeostasis. So these tissues arise through the 3 germ layers. So during development, these 3 germ layers give rise to tissues and organs of the animal embryo. So the ectoderm, endoderm, and the mesoderm are the 3 types of germ layers. Diploblastic taxa only have ectoderm and endoderm. Triploblastic taxa have all 3, so they have ectoderm, endoderm, and mesoderm. Mesoderm is the intermediate, so it's the middle. So ectoderm comes up top, mesoderm in middle, if they're triploblastic. If not, endoderm will proceed next. But we're talking about, we'll put an example of triploblastic animal. So ectoderm on top, mesoderm in the middle, and endoderm at the bottom. So the mesoderm gives rise to the muscles and most other organs. Ectoderm gives rise to the integumentary system in animals, and also the central nervous system in some. And the endoderm is the innermost germ layer, and it gives rise to the lining of the digestive tract or cavities and organs. So body cavities. Body cavities is either a space or filled with fluid. A true body cavity is called a coelom. So here we have a coelomate, which means that they have a true body cavity. A true body cavity is when the space is surrounded by mesoderm. So here we have the space right here, and it's completely surrounded by mesoderm. Now let's compare it with the pseudocoelomate. Here we have one continuous space, it's not surrounded by mesoderm. It's surrounded right here. Yes, it's surrounded in a ring, but this is completely surrounded, meaning it's surrounded on two different levels, one to the left and one to the right. Well, a coelomate has no body cavities. The only cavity is the digestive cavity. So it's basically filled in with mesoderm. So a body cavity has many functions. First, it has fluid in them, and it suspends organs. And when the animal falls or has an impact, that fluid absorbs the impact and cushions the organs. So it's kind of like a pad, almost like a protective pad around the organs. And the fluid acts like a skeleton against which muscles can work. So the fluid, as in worms, worms do not, I believe they do not have bones. So how worms move is that they push, their muscles push against the body cavity in order for them to move. And a cavity enables internal organs to grow so they can grow and move independently from the outer body wall. You don't want, you don't want an organism with all the organs tightly squeezed together. You want their space and their wiggle room. Because if a body, for example, let's say the liver becomes inflamed and it gets all large and it starts putting pressure on your other organs. We don't want that. We want the nice space. We want space for each organ or some organs. And we'll be talking about protostomes and the pterostome development. So based on early development, many animals can be characterized as having protostome development or pterostome development. These developmental modes differ in cleavage, coelom formation, and the fate of the blastopore. We'll be only talking about the fate of the blastopore. So let's just go briefly over this. So cleavage in protostomes, it is spiral and determinate, and to pterostomes, it's radial and indeterminate. So you see how these cells are arranged in this one? It's like a knob, it's like twisted. So these cells are arranged differently in cleavage. Coelom formation. So in the protostomes, the solid masses of mesoderm split and form the coelom. So just imagine like a piece of cells right here, they form their own space, while in the pterostomes, this layer of cells will pinch out. You see they're being pinched out this way, they're being pinched out, and using the space of the archipteron to form the coelom. But we're only going to be talking about the fate of the blastopore. So in protostomes, the mouth develops from the blastopore, while in deuterostomes, the anus develops from the blastopore. So this is the digestive tube, and this is what animals are characterized as having protostome or development or deuterostome development. So zoologists recognize about 36 animal phyla. And they have done this by using tools like ribosomal RNA genes, Hox genes, protein coding, nuclear genes, and all these types of genes. And what they did is they just put a table and sort these animals out, which ones are related, which ones are distantly related, which ones are completely different. They just organized these animals. So here we have animals. Well, it's not 12 dozen of, that's not the 36, this is only like a fraction. But here we have all animals come from an ancestral protist, as we know. So we'll be talking about this with the slide. So there's 5 important points about the relationships among living animals are reflected in their phylogeny. So all animals share a common ancestor. Of course, it's the ancestral protist, or choanoflagellates. So here we have the split, and then they split again, and they form this huge tree. Sponges are the sister group of all other animals. So sponges, right here, I think it's Porifera, and there's the sister group. Sponges, Semnodophora, and Cnidarians do not have bilateral. They don't have not bilateral. They don't have no type of symmetry at all. They're asymmetrical. And I think these guys, Echinodermata, don't have symmetry either. I wonder if they don't. I think they don't. But they do fall under the Bilateria. That's a good question. I'm gonna look at that later. I'll have to write that down. Hold on. That's a good question. I'm about to look at it. Do sponges have symmetry? But they're part of the Bilateria. Huh. That's a good question. I'm not gonna think about that. I'll think about that later. The Eumetazoa are the true animals, is the clade of animals with tissues. So true animals have tissues. So Eumetazoa, right here, have true tissues. I'm lost. I just got lost for a minute. Okay, so most animal phyla belong to the clade Bilateria. So here is all the animals in Bilateria. So I think these should have, yeah, they must have bilateral symmetry. So I just organized these so you can understand it a bit better. There are three major clades of bilaterian animals, all which are invertebrates, animals that lack a backbone or a spine, except Chordata, which includes vertebrates, animals with a backbone. So we are part of Chordata because we have spines or backbones. So these are the three clades: Deuterostomia, Lophotrochozoa, and Ecdysozoa. Ecdysozoa. So these are three clades that I circled. 1, 2, 3. Platyhelminths through Arthropoda have no spine. Chordata have spines. And Hemichordata, Echinodermata have no spines. Or invertebrates, and vertebrates. I'm still trying to figure out why I circled this. So whatever I said regarding to these, don't pay attention. I'll put in the description or something. But thank you for watching this lecture.